Today, I'm going to share a little bit about the decline and the ongoing recovery of this really iconic frog. It's beautiful. I try to include as many really good photos as I could. And really, our story starts with this global biodiversity crisis. And like I mentioned before I started sharing the presentation, most conservation is talking about focusing on like what is happening, like where are all these species going, how many are declining. But what I really like to think about is how can we stop that decline and reverse it? What are the methods that we can use to find conservation success stories and be able to share those stories and perpetuate them? So, under the Endangered Species Act, whenever a species is listed as threatened or endangered, these recovery plans are drafted, and they can include a number of different methods depending on the species' life history and the threats that are currently facing, or that the threats that the species is facing. And some of these methods, for a really small population, might be captive breeding. Usually, there's always some component of habitat restoration that might involve removal of invasive species. A huge component is public education, which is why I, as shy as I am, try to always say yes to these public engagements because I think it's really important to spread knowledge and share that with others. And then obviously the focal point of tonight is these translocations, which are honestly an up and coming conservation strategy. So translocations are a really important tool for population recovery, such that they're actually written into 70% of the recovery plans for endangered species in the United States. And of those species that have been delisted, 30% of them use translocations as part of their recovery plan. So it really is up and coming. It's really common. You probably have heard of some translocation efforts, especially being, from state parks or whatever park you're working with. You probably know someone or currently work on a project similar to the one that I'll be sharing tonight. However, translocations are really far from easy. So there was this review done a while back that tried to identify all of the different difficulties that translocation efforts might encounter, and they estimated over 1,200. So this is out of just under 300 translocations, and they tried to really boil those different difficulties into these broad categories. And I'll mention three of them that I think are most pertinent in the framework that I'll try to put tonight's talk into, which are the intrinsic factors. These are the sorts of elements of the animal that you're trying to recover. It might be their behaviors or their general life history strategy. It could be loss of genetic material within that species that you're trying to recover, which limits its evolutionary potential. It could just be a really cryptic species. You don't know how many are left. It's hard to find them, to translocate them, and it's hard to monitor them once you've moved them. Or it could be something like disease or disease resilience. Another broad category that these difficulties might fall into are extrinsic factors, which are characteristics of the environment where you're trying to move the species or repatriate an area. And that could be like the quality of the habitat, which is often degraded from, previous status when the species was still occupying that area. It could have transformed vegetation communities. It could be completely paved over and urbanized. It could contain novel pathogens. And as always, there's some element of climate change that could be affecting these environments. And these are sort of the first factors that we look at when we're selecting translocation sites to reintroduce a previously extant species into. The third set of factors that make translocations really difficult is that they're really expensive and they take a ton of work. So just in funding people like me, a biotech at Santa Monica Mountains for 8 years, that's quite a bit of money that went into this frog project. And that's, I'm just a peon of the whole crew. So there's a lot of money that goes into the recovery of these species, which is why, it's always easier to just maintain our biodiversity than to try to recover it. Even so, there are examples of translocation success, and I really like to highlight these because it's our inspiration to keep doing the work and to keep hoping that we will be able to see red-legged frogs again on our landscapes. So here is not a red-legged frog, but a mountain yellow-legged frog, which I think is a really fantastic example of a translocation success. So looking a little bit closer at the data from this is a recent paper that was published showing translocation data from the last 10 years effort. And here we can actually start to see the different factors that might be at play in translocation success. So on the y-axis, we have frog survival. Did the frog actually survive a full year in that mountain lake? And we have varying degrees of frog survival depending on which habitat it was reintroduced into. And that might be due to like presence of different pathogens in those sites, or it might just be some lake basins have more nutrients or more food. But we also see a pretty like strong effect of the donor population on that frog survival. The green donor population had really high survival rates. And that just shows that there might be some sort of evolved resistance, for example, to a pathogen, or it could just be like one genotype is a little bit more superior than another in just having survival for that year. And obviously, survival is just one measure of success. Ultimately, you would want a translocated site to have self-sustaining population. So we would expect to find egg masses, them breeding on their own. But this is just looking at that first pass. Did the frogs even survive? Now, as many of you may know, mountain yellow-legged frogs are kind of the flagship species in California for chytrid decline. So that's why I kept using disease as an example, because this species really did face really precipitous declines in response to chytrid fungus, which is a global pathogen that's affected amphibians. So when we're considering these intrinsic and extrinsic factors of success, we really also need to put that within the framework of like a history of disease and whatever other factors these frogs might be facing. Now, a little bit more background about the chytrid fungus, because this is one of my major focal questions for my dissertation work. Now, it's implicated in global amphibian decline. It's caused extinction of many species. And looking a little bit closer at the genetic composition of this pathogen, there are two major global lineages that are really pathogenic. And in North America, both of these lineages can be found. And something that is important to note is that these two lineages, GPL1 and GPL2, can form really close association species. So that means that if you have two species of frog living in the same environment, you might find GPL1 infecting one species, but not the other, and GPL2 affecting that species, but not the first. So this kind of is evidence that there might be some sort of coevolution happening where frogs are like more susceptible to non-local lineages. And they've tested this in the lab, and they found that when you take a lineage that isn't present in an environment and introduce it, then there are actually higher mortality rates. Mortality rates. So what this is really saying is that we should be concerned about spreading this pathogen, even if you find the pathogen present in somewhere like the Santa Cruz Mountains. The introduction of a different lineage from, let's say, the Sierra Nevada could actually increase mortality rates on whatever amphibian species are present there, even if they've developed endemicity and resilience to the local lineage. So this is just kind of the framework that I'm approaching studying chytrid fungus in something like a red-legged frog, knowing that even if you're not seeing these mortalities, it's still an implication in their survival to know like what pathogen is present and how can we limit the introduction of more pathogens. Okay, and then the last thing to note about chytrid fungus is that susceptibility to the disease varies not only across species, but also across populations. So some populations may have evolved greater resistance than others. Now, I said all of that really because the California red-legged frog is really resilient to chytrid. They actually, most populations have the pathogen present, but not too many mortalities have been observed. And as promised, a little bit of life history background about the California red-legged frog. This is the largest native amphibian in California. Keyword native, because unfortunately bullfrogs are present in California and they are a little bit bigger, but they are massive frogs. This is not even the biggest frog that I caught. The biggest one was over 300 grams, which I promise you is just, it's obscene. It is so cool how big they get. They were actually consumed really widely by miners during the gold rush, which was potentially one of their first causes of decline in the late 1800s. They were also popularized by Mark Twain. Some of you may have heard of this story he wrote, which actually helped him rise to fame, called The Celebrated Jumping Frog of Calaveras County. They all believe that this story was written about this frog, and it was actually what helped this class of elementary school students finally get the governor to agree that the California red-legged frog should be our official state amphibian. This was an effort that was done by the organization Save the Frogs, which really helps with conservation efforts, just so that people know what this frog even is and know that it exists. So the California red-legged frog has a really classic frog life cycle. They breed for only a couple of months each year, and the timing of breeding is really dependent on the latitude. So frogs in Baja California are probably breeding between December and January. They're probably wrapping up their season right now, whereas the frogs in Southern California will start breeding next month. Not so sure about the frogs in the Bay Area. They're probably even a little bit later, depending on the rain this year. Now, each egg mass can contain up to 4,000 eggs, which is insane. They hatch about 2 weeks later, depending on the temperature of the water. And what's really interesting is frogs at these southern latitudes often overwinter. This has been observed in a few populations, including the one that I've worked on, where the tadpoles just get absolutely massive, and then they just stay in their tadpole form until the following spring when they sprout their little legs and carry on with their lives. Very interesting. Nobody has really looked into what that cue is. And then the last thing I wanted to mention in terms of their natural history is that adults have a really strong homing instinct. So when you think about something like a translocation and the intrinsic factors at play in success, we can't move adult frogs from a population because they will probably just try to get back to their natal site. So all of the translocations for red-legged frogs use egg mass translocations, where we harvest partial egg masses and we'll introduce them to a new site. Here's a video of one of the tadpoles that we released. This is not even as big as they get, and it is probably 4 inches long. So unfortunately, California red-legged frogs range has declined significantly. They were listed as federally threatened in 1996. And you can notice that most of the Central Valley used to have these frogs. Unfortunately, the wetlands were all drained, and a lot of that area is now used for farming. And the remaining ponds actually are really good habitat for this frog. But farmers and landowners don't want to have this frog on their landscape because it really inhibits the work that they can do. So often those ponds are drained and then the frogs just blink out. There are arrows pointing at the northern and southern ends of their current range, because typically with range contraction, those are the populations that are most susceptible to decline. There just isn't as continuous habitat for them to survive, and there's a loss of the metapopulation structure. I will Oh, yeah. I'll walk you through some of the putative causes for decline, and then I'll provide some case studies on the translocations that are currently underway in California. So, as I just mentioned, habitat loss is probably the biggest cause for decline. As you can see, like California is largely developed, and that just inhibits the, remaining habitat for this species. There's a whole laundry list of other possible reasons. Interestingly enough, disease always has a big question mark next to it because they are really resistant to chytrid fungus. But there's really not like a clear answer whether like earlier declines were due to this disease and the remaining populations have evolved resistance, or if they were just always resistant to chytrid. So more work really needs to be done on museum samples to get at that answer. Poaching, is in reference to the 49ers, and San Francisco used to serve up red-legged frog legs pretty commonly in their restaurants. So that may have had an effect in the 1800s. And invasive species is a big risk factor currently for their decline. And I'll talk a little bit more about that in a second. So while we have this question mark next to disease, the first documented mortalities due to chytrid fungus were in 2020. This was actually in the source population for the Yosemite translocation. And what was crazy about that was this was one of our collaborators who published that paper. And that same year, we started documenting mortalities due to chytrid fungus in our source population. So this was 2020, and right about the time that I had started talking to my current PhD advisor, which really was like what encouraged me to go on and get my PhD and really look at this question. Like, should we actually be worried about chytrid in red-legged frogs, or are they as resistant as we think? So as I mentioned, these translocation efforts are currently being conducted throughout the state. I know Golden Gate has a project underway. Yosemite Pinnacles had a project. They were the first ones in the early 2000s. There is another effort up in Napa. There's the one that I've worked on in Santa Monica Mountains. And then there are additional efforts down in Riverside and San Diego County. So just to provide like a little bit of a case study using this framework of the extrinsic factors, such as non-native species removal and habitat recovery, I'll talk about the work that's currently being done for these translocations to limit the effects of those factors. And then I'll talk about what I would like to study in terms of frog genetics and disease resilience and how that might be inhibiting translocation success for this species. So in Pinnacles, there's this long history of fish introductions in their reservoir where frogs used to be really abundant. And in the late 1980s, they began to use electrofishing to just get rid of, or actually they just drained the reservoir to get rid of all the bullhead catfish. And then in the 1990s, someone had introduced green sunfish, which just completely exploded. And so they had to do electrofishing to get rid of all of the green sunfish, which is a beautiful fish pictured in the middle here. But this site, once it was cleared of these fish, the frogs did extremely well. They introduced their first egg masses in 2001, and within 3 years, they had the frogs as adults already breeding on their own. And this was really the inspiration for the translocation that is currently underway in the Santa Monica Mountains. And it's actually laughable, like how effortless they made this seem, because we thought it would be as simple as like, take these egg masses, add them to the water, and then suddenly, boom, you have like breeding frogs. But it's really much more complicated than that. I can definitely believe the 1,200 difficulties estimate of that review paper. But yeah, so this is just a really good example of management trying to take control of those extrinsic factors that are limiting frog survival. These fish predate on the eggs, so it's really important that you get rid of those non-native predators. Okay, and then what's really amazing is that in Yosemite Valley, they were able to get rid of all of the bullfrogs that had been introduced which is really, really challenging management work. And their reintroduction started in 2017. I personally haven't heard any media about success of that project yet, but hopefully we'll be able to see red-legged frogs in the valley and mountain yellow-legged frogs up in the high elevation lakes like soon enough, because that's the way it should be. That's the way it always was, and I think it will make a visit to that park like even more stunning to see the native wildlife. Okay, and then the last sort of little case study that I'm really proud of is some really good friends of mine, collaborators, have been part of the first ever binational translocation. So these frogs in their first year were actually, well, the egg masses were helicoptered from Baja California, went through customs, and were eventually found their new homes in San Diego and Riverside County. And this project, so far, they have had some success, such that they have evidence for survival. But when considering those factors that really limit monitoring ability, these two sites are watersheds. They're not ponds. So this really just opens up the possibility for frogs. They're moving, living creatures. They will move up and downstream. And unfortunately, downstream from their introduction site is private property. So being able to monitor survival of these egg masses, now frogs, is really challenging and has really inhibited their ability to say how successful this project is. But so far, they've had, I believe, 3 years of translocations, which I think is phenomenal considering all of the constraints of working with 2 countries to do this really important conservation work. And I'm sure some of you might be wondering, why the heck are we moving frogs from Baja California up to California, San Diego and Riverside, when we have this nearby Santa Monica Mountains population here? And again, like looking at the intrinsic factors that we consider in frog survival in these translocation sites, preliminary DNA work was done that showed that there's actually a pretty strong genetic divide right here in the LA basin. So interestingly enough, the frogs from Baja California are more genetically closely related to the frogs that used to be in San Diego and Riverside, which is why they are instead choosing to move these egg masses 420 kilometers to the north to repatriate this landscape. So hopefully they will continue to see frogs and hopefully egg masses in these stream sites. But I can attest to how difficult it is to conduct frog translocations in a stream-based, site. And for those of you on this call who may or may not be interested in conducting a red-legged frog translocation, I would say if it's a pond, like, carry on. Like, that is awesome. You're going to have a lot of success. It is much more challenging with the streams. So Santa Monica Mountains is, my home, my site where I have worked for 8 years on this translocation. And I'm excited to share a little bit more about my experience on that project. So here, some of you who joined like the horned lizard talk might recognize this area because this has been where I've done all of my field work for my master's and now my PhD. But this is our source population up here in the Simi Hills, which is this area just north of the 101. This is a major freeway. And then frogs were last seen in the Santa Monica Mountains in 1970. So our goal for this project is really to bring back the frogs to Santa Monica Mountains. Now, there have been a number of successes and setbacks for this project over time. And, the first success was that we moved these egg masses and they hatched into living, breathing tadpoles, which is already remarkable. I would count that in a success column for sure. Those tadpoles grew into frogs. A-OK, still going great. Those frogs laid eggs. Honestly, everything was a little too easy at this point, and I joined the project in 2016, right at the peak, when everything was amazing, and I was so inspired by these frogs. Shortly thereafter, the entire park burned, every single frog site, including the source population. With that, we had limited ability to conduct work together in the field due to the global pandemic. And the subsequent sedimentation and erosion due to all of these aseasonal storms and flash floods that occurred over the next 5 years made the habitat recovery really fall into this boom and bust cycle where we would go into the field and I'd be crossing my fingers and it would look like a bowling alley, completely filled in with mud, just no deep pools, which these frogs really need to survive. And then I would go the next year and they would be completely cleaned out to bedrock. So it was really hard to judge whether or not the frogs would still survive, whether these sites would ever be salvageable. And, add on to that, that we just had a second fire burn our source population. So this project, I would say, has had a lot happen in the time that I've been there. Never would have expected a lot of these sorts of big setbacks. But all of that said, there have been wild successes. Not only did we see breeding in all 4 of our translocation sites, but one of those, which we always deemed our golden child, has had 8 consecutive seasons of breeding on their own, which is, I would say, like, that's the goal of the project. So that is huge. Like, cheers to that. And in addition, hopefully that number will bump up to 9 this year. But last spring, we also discovered this sort of bonus, like, secret population that had come back since that 2018 fire. And there are like probably 20, if not more, adults surviving and reproducing in that site. And we have evidence that this is the 3rd consecutive year that they have bred. So it's possible that 50% of our translocation sites are actually hosting populations of red-legged frogs now, it's just massive. I cannot stress how amazing that is, given all the tears that went into this project by not just me, but my boss, who was there setting the groundwork. So naturally, as a PhD student who needs to have 3 chapters rather soon, I'm very interested in looking at these intrinsic factors that might be limiting success of these populations. What's going on in those other 2 sites? Obviously, there's some extrinsic things at play, like with the habitat recovery, but are there genetic factors limiting this? So just to remind you, these are the sorts of intrinsic factors, just a few. There are many more that I'm sure that us all as a group could come up with. But, I was always curious, number one, whether there were mortalities happening that we weren't seeing, because amphibians, unfortunately, will just degrade like really quickly. Like once they die, they decompose and you just don't detect that loss. But as someone who has always studied population genetics, I've also always been really interested in genetic diversity. So obviously, we're taking these frogs from this one site and artificially selecting a few egg masses and subsetting that genetic diversity in these 4 other sites. So how is that affecting that population's ability to sustain itself? And what effect does that have on like the adaptive potential of that site? How well will it be able to handle environmental change? So today I'm going to talk a little bit about the disease work, because that's what I've been working on the longest. I have some preliminary data to show in terms of chytrid in the Santa Monica Mountains, and then I'll introduce my second project. So my primary goals for this first part is to determine, like, is chytrid fungus present? Chytrid fungus is BD, that's just the scientific name. How much is it? How many individuals are infected? And then of those infected individuals, how infected are they? So it's a little bit like taking a COVID test. Is it a yes or no? Do you have it or do you not? And then how severe is that infection? That is the load. My second aim was actually to figure out what genetic variation is present. So back to those 2 global pathogenic lineages, I want to know, is it just a single lineage? Do we have both? What do our sites really contain? And how can we maintain that variation and not introduce new variants? So to do this, I collected a bunch of swabs from five sites. The donor site, which is our source population, as well as the four translocation sites. And I sampled all of the amphibians present. This kind of goes back to that first slide about chytrid fungus and how it can have a very specific tie to different species. So I was curious about the pathogen in its entirety in these sites. So I actually looked at both species of tree frog. We have the Baja California tree frog and the California tree frog, as well as the red-legged frog and the California newt. I use really standard methods in the lab. I'm happy to discuss more with anyone who wants to nerd out about lab methods, but I assume this wasn't the audience for that. And then I used these two different methods to figure out whether it was GPL1 or GPL2. Also happy to discuss that with anyone who cares. So my next step will be to use linear models to look at how these pathogen loads that I detect are influenced by different environmental variables, which is something that I'm currently working on, and unfortunately don't have preliminary data to show, but that will be, I guess, at the next talk that I give to Henry Cowell, because this seems to be an annual thing. Okay, so diving into the data, what we first see is pretty high percent positive infection rates, which means that of all, I'm lumping all four species here, just at a first pass, we have nearly always over a 50% infection rate, including an 86% in 2023. I'm still processing samples from 2024, so unfortunately, I don't have those data yet. But another really interesting immediate find is that like this number, this prevalence jumped 50% between 2022 and 2023, which in conversation, I've talked with people from Santa Barbara and other people who are working in the Bay Area. They have seen a similar jump in pathogen prevalence across that time frame. So I'm really interested in knowing exactly what was going on to increase the spread and the infection rate of this pathogen of amphibians in these areas. When you look at exactly how infected those positive individuals are, there's also this really interesting trend where in 2022, when the percent of individuals was lowest, they're also like the individuals that had the fungus had very little of it. So it's almost like in 2022, this pathogen was hit hard for some reason and had like a massive rebound in 2023. Very interesting patterns falling out that obviously I will need to control for seasonality and a lot of other potential environmental factors. But we can see that this high prevalence and pathogen load suggests that these animals are existing with the pathogen present and they're surviving. None of these animals that I swabbed looked like they were on their way out. They weren't sickly or anything. They are pretty much surviving, coexisting with this pathogen, which is fantastic. It's one less thing to worry about in terms of, should we be eradicating chytrid? Maybe not. Maybe they're okay. The maximum load that I found in my study was in a red-legged frog, which kind of goes with this belief that red-legged frogs are really resistant to chytrid fungus. And as I said, we're really going to need my models to figure out why these trends are popping out in the data. But since I don't have those to show you, I will split out the data based on species so you can get an idea of some of the patterns that are kind of hidden within the data that I just showed. So when we separate the percent positive infection based on species, you can already see a pretty big difference between red-legged frogs and the Baja California tree frog and these two other species, such that these species are carrying like a higher infection rate. Most of them or 50% or more have BD on their skin. Similarly, they're carrying much higher loads of this pathogen, far higher loads. And what's really interesting is that even though 34% of newts were positive for this pathogen, they were carrying a very, very small amount of the pathogen. So what's interesting about this is that we're really starting to see that these two species here have a very different strategy for handling presence of this pathogen, such that these two are likely much more tolerant of the pathogen. They can handle infection. It's not really inhibiting their survival. Whereas these two species are more resistant. They're just avoiding infection altogether. And this might be due to some difference in behavior. Maybe they're less aquatic. There's something else at play that's helping them keep their percent their infection rate low. And also when they have an infection, keep that very minimal. Okay. And then some preliminary results of the sequencing that I did. I found this really fascinating result. I was really not expecting this, where the source population up here in the north only contains GPL1. And that's using both methods of sequencing that I conducted. There are over 100 samples included in this map, just north of the 101 in that site, which means that it's very strange that there is not GPL2, whereas far fewer samples, as you can see in our translocation sites, and yet I picked up both lineages. So obviously, I assume you're all panicking now, right? Because we're taking egg masses from this site that has only been exposed to one lineage, GPL1. I just told you earlier that amphibians have a higher mortality rate when they're exposed to a novel lineage. And we're exposing them to a novel lineage knowingly. We're taking them and moving them into this site that has both GPL1 and GPL2. And interestingly enough, we have observed some frog mortalities, not only in one of our translocation sites, but also up in the source population. However, this site has had 8 consecutive seasons of breeding. They're clearly surviving. They're clearly able to function. And we have that additional site that has 3 seasons of breeding. So I would say we don't really need to be panicking right now. We should proceed with cautious optimism. This result really made me feel like I wasn't completely wasting my time on my dissertation, which is something that I think every grad student wants to feel at some point in their career. So that was really cool. And I have plans actually in about 2 weeks to go back to Berkeley to fill in this sampling a lot more to confirm what this difference in pathogen genetic variation really is. So more soon on this, but definitely a really interesting result. Okay, are we doing on time? So the last thing, this is really just a preview of my next chapter, which is going to be looking at the genetic effects of translocations. I've been so fascinated by this concept. So in general, I have frog samples. I actually collected buccal swabs by putting a swab in each of their mouths, like a lollipop, and just collecting a nice DNA sample. And I will estimate genetic diversity, genetic load, and inbreeding in each of these populations. And what this is really going to be used for is to figure out how much of an artificial bottleneck we're putting on these populations and the impact that might have on adaptive potential. So the environment is changing. It's going to keep changing. These frogs are facing new and novel environments, and I want to know whether or not we are setting them up for success genetically. What's interesting about small populations is that even though the classic idea is that low genetic diversity is always bad, it's not always the case. We're actually taking egg masses from a pretty small population, which has been small for a pretty solid amount of time. There are probably about 100 to 200 frogs in that site every year for like the last 10 years. And because that population has remained small, it's likely that it has also been purged of any harmful mutations. which means when we're subsetting that population and taking genetic material and moving it into the Santa Monica Mountains, it's unlikely that we're taking deleterious mutations, anything that will cause severe illness, and moving them into these sites. So this actually enhances the ability to have translocation success. Okay, so overall, especially all of the work studying chytrid fungus, I think that this work was extremely important in developing proper biosecurity measures. We weren't doing any sort of bleaching or any sort of biosecurity prior to learning more about chytrid fungus, and I think that it should be just, the first step of every amphibian project, given how prevalent this pathogen is. We can also use these data to track pathogen introductions and changes over time, which could help us explain any observed mortalities or changes in survival, and hopefully improve, persistence of these translocated populations. If I can get through that second genomics chapter, I'd like to understand the genetic impact of translocations as a recovery strategy. If we could set some sort of threshold of number of reintroductions we need to undergo in order to have the level of genetic diversity observed in the source population, then at least we can remove the impact of low genetic diversity on survival of the population. And then altogether, as I've said, I'm really interested in these intrinsic factors because I think that we always focus so much on rehabilitating the environment, and I want to know what can we do about the frogs themselves to enhance their ability to survive, and how can we integrate this into our management plans to be more adaptive and anticipate, the genetic factors that might be at play. Okay, and hopefully I'm good on time to answer some questions, but I first want to thank everyone that has helped in the field. This is all National Park Service work. I'm currently no longer working with NPS as of this season, which is very strange. I've turned to data analysis, so hopefully as time goes on, I'll have more stories to share as I actually parse through the data. But it's been absolutely phenomenal getting to work on this frog with all of these amazing people. And yeah, next time that I'm in the Bay, Dylan, I really do want to come visit Henry Cowell. I'm sorry that I couldn't be there in person today, but I'm happy to take any questions you guys might have.